Oh, kia ora, folks. This is I'm Martin Kennedy, for those who don't know me. It's a real pleasure to introduce our speaker for today's Genomics Aotearoa seminar. Thank you for coming along. Our speaker is Professor Sarah Medland, who's visiting Christchurch on a Margaret Black Fellowship. So she's with us at the School of Medicine, Otago School of Medicine here in Christchurch. Sarah flew in last night and she's already given one talk, and this is her second of the day. But she's has very strong Kiwi roots, although she hails from Brisbane. She did most of her training in Brisbane, BA Honours and a PhD, and then postdoc in Virginia for several years. And then she's been based back at the Queensland Institute of Medical Research for the duration, I think. Sarah's a psychiatric geneticist, psychologist by training, but really has been making a lot of inroads into psychiatric GWAS and understanding of complex mental health and neuropsychiatric conditions. Sarah's a fellow of the Academy of the Social Sciences in Australia. She's also a member of the Australian Academy of Health and Medical Sciences, and in 2000 she was awarded the Order of Australia Medal on the Queen's Birthday Honours List for services to medical research in the field of genetics. Sarah's published over 475 papers, H-index of 111, and which sounds like a phone number, but it's nevertheless very impressive. And her work has had over 50,000 citations. So she really is a leader in the field, and it's a real privilege to have Sarah. So if you have questions as we go along, please pop them into the chat and we'll pick those up at the end. Meanwhile, it's my pleasure to hand over to Sarah. Thank you.
Okay, so thank you very much. Just give me a moment while I share my screen and get started. Yes, and I'm sort of sufficiently embarrassed by my introduction, so yeah. It's a great pleasure though to be speaking with you, and I'd really like to thank Martin and University of Otago for bringing me over. Okay, can everyone see my slides properly? Is that working? Yep. All right.
All good, Sarah.
Oh, great. Okay, so I'd like to start by just acknowledging where I'm coming from today and where I'm talking to you from. So I am here in Christchurch, and acknowledging the traditional custodians and the owners of the land, and paying the respect to the the culture and the ongoing living connection to the land here. But also in Australia, so where I work in Meanjin, which is Brisbane, and I work on the lands of the Turrbal and the Yagara people. And so, sorry, that arrow shifted. It is meant to be in Brisbane. Okay, so I don't have any conflicts of interest in the commercial sense. As Martin mentioned, my training's in psychology and genetics. And so what I'll be talking to you about today is really around where we're at with mental health genetics, and I'm going to use the examples of major depression and bipolar disorder. Depression being very common within the population, bipolar disorder less common, but still fairly frequent. And just using these two as examples of how things are working in the mental health genetics space. I am originally from New Zealand, so my family moved to Australia when I was 7. Yeah, so sort of Central Otago and those regions are what I consider as home for me. Okay, so I know a lot of you work across fields, but you will have heard quite a lot about GWAS. So just for anyone who is less familiar, we usually explain this as a group of cases and controls. We collect a cohort of people. We collect phenotypes on these people. We need a DNA sample, which these days is usually saliva. And then we tend to use a chip-based genotyping method. So this is the SNP chips, or the GWAS chips as they get referred to. Those chips have about 500,000 data points that we directly genotype, and then we take that, we combine it with the public reference sets such as the 1000 Genome or the it used to be the HapMap. TopMed is currently very popular. And from that, we will get on average around about 9 to 10 million usable data points after imputation. We take that forward for GWAS analysis, and when we work with that imputed data, we are analysing the dosage, so it takes into account the uncertainty around the imputation. So we don't reduce it to hard calls. We work with that full range of spectrum. And the analyses we run for this are mostly regression-based. We do correct for relatedness and the effects of known confounders and important covariates, but effectively it's a regression technique, and we tend to brute force this across the genome and then go back with a more nuanced model in areas where there's signal. So what can we do with this? Obviously, the knowledge generation is one of the key aspects. But one of the important things from a mental health point of view is that most of the effects we are finding, similar to other complex traits, they're very small effect sizes, so they're not really clinically actionable at the individual level. But nevertheless, this technique is really cost effective. So it's around 100 to 150 Australian dollars to collect a sample, take it all the way through to genotyping, and including genotyping in that cost. So we think this is a fairly cost-effective way of doing large sample work, which is important because our effect sizes are very small. So what I'm mostly going to be talking about today is polygenic risk scores or polygenic scores. And so I've just got a couple of slides here for anyone who hasn't worked in this space around building predictors. So we go ahead and we have a genome-wide association study of the trait that we're looking at, and the example here is height. And we go in and we look at what are our top findings, what are the effect alleles, what are the effect sizes, and we start to build up a weighted sum score. So for this location, we have an AG SNP, and everyone who has a G becomes magically 2 centimeters taller. So people who are GG are 4 centimeters taller than someone who's AA, for example. Obviously a false example, but just to get the idea of how it's going to happen, we can go in and we can add that weight to the person's score and then go ahead and look at the next locus that we want to include, and so on all the way across the genome. And so what we get is this weighted sum score, and it'll give us an estimate of our polygenic risk or the biological risk for the trait. We then usually take that forward for a regression-based analysis once again. And here we typically are looking at how much of the variance in the trait we can explain, or how well we can predict. And so that's all fairly straightforward. And so just using the example of depression here, one of the important things to know about the power of our polygenic risk score analysis is it's actually the size of the discovery GWAS sample and the accuracy of those weights that are the limiting factor on that power. So it's not really about the size of the sample we're predicting into, it's more about the size of the sample that was included in the GWAS. And so one of the key points here is that, you know, as we increase the sample size, the prediction will get better. Here, what I'm showing on this slide is prediction into the same data set. So this is, these two regressions have been run on exactly the same data set. The difference is, though, that one predictor was built from the first generation of the Psychiatric Genomics Consortium depression analysis, which had a sample size of around 18,000. And the second one is built from the generation 2 of that analysis, which has a substantial increase in sample size and then a substantial increase in power. And so we're at the point at the moment where we can predict around between 5 to 10% of the variation in a trait. For clinical work, that's really not good enough, but for research purposes, that's a really strong predictor. So that predicts, for example, more than age or sex alone. One of the other major problems that we have with the work that's being done and the polygenic score analyses is that they are heavily dependent on the LD structure, so the autocorrelation structure within the genome. And what that means is that they perform best in a prediction sample that best matches the discovery sample in terms of ancestry. And so because most of the genome-wide association studies that are out there are being run with European ancestry people, that means that we are currently better at predicting within that ancestral group than within other groups. This is an unfortunate fact. People are working on trying to both increase sample sizes for other ancestries to have bigger discovery samples in those ancestries, and also try and work out if there's any way that we can ameliorate this with statistical analysis. But at this point, we don't have a way around it. It's out there. But where this starts to become really important is if we're starting to think about using these kind of risk scores in clinical practice or clinical medicine in any way. And so we need to be aware that the use of these scores at the moment would come with an intrinsic worsening of health equity for non-European groups. And that's a major problem. So although we can look at sort of case versus control analyses, we can also look at how the risk increases as you start to get more and more of these risk factors. And here, this is a polygenic risk score that's been broken up by decile, and then looking at the risk of having a depression diagnosis by decile of polygenic risk. The sample here is from the Australian Genetics of Depression study, which I'll show you just in a moment. And there's two different predictors, once again, very similar, built on GWAS of major depression. And as you can see, as you start to increase in terms of decile of risk, your odds ratio of having a diagnosis increases. So what can we do with this kind of thing? Obviously for research, it's really interesting and very valuable. It's a great predictor, and it's starting to have enough power to be really useful in the research space. It can also potentially be useful in a family history situation. So particularly where the person isn't in touch with their family, or there's no one in the family who has accurate information. And that could be because the family themselves don't know, but it equally can be that, particularly for something that's a late onset, potentially the diagnosis that actually is running in the family wasn't known when the older people were being diagnosed, maybe 20 or 30 years ago. So it can really help when there is uncertainty about what a diagnosis is. Often the family will know something happened with this person or something happened with this person, or they grew up in care, but they might not exactly know why. And so it could be useful in those kind of situations if the family is looking for some knowledge or certainty, or want information about a younger person in the family who has a diagnosis. And so one of the big issues is, can these or should these be used in diagnosis? And I think the answer really depends a little bit on the trait that we're looking at and the type of medicine that we're talking about. So for some things, yes, probably. But for mental health, I think it's a bit more complicated, and I'll come back to that in a few minutes. Okay, so just to give you a quick overview of where we're up to in the GWAS space for depression and bipolar. So most of this work is done through a consortium called the Psychiatric Genomics Consortium, and I know that Patrick Sullivan, who has been very much a leader within the Psychiatric Genomics Consortium, has presented to this group as well. So I don't want to sort of rehash any of that. But basically, what these consortiums do is bring together samples from across the world. And so it basically gathers in as much data as possible and as many investigators as possible. And so these papers end up with 300 or 400 authors, which has a whole series of complications in terms of what their CVs look like, in terms of how their effort is understood, and in terms of how journals treat large authorless papers. So I'm happy to answer any questions about that later on. Okay, so just to give you an idea of the journey. So depression, for example, our first big GWAS from the Psychiatric Genomics Consortium was in 2012. And at the time, this was pretty big. This was 18,000 people. It was a lot of work to get those people together. And they'd all been really carefully interviewed and assessed by clinicians, mostly psychiatrists. Unfortunately, the 18,000 people did not yield any significant results. The next thing that kind of happened in the space was 23andMe, which is the direct-to-consumer company, basically did a questionnaire, and they included one question. They asked people, have you ever been diagnosed with depression? Yes, no, I don't know. They took the yes as case, they took the no as controls, and they dropped the I don't know. And when that data was put into a GWAS and then meta-analyzed with the PGC data, all of a sudden we had new signal. So we have new significant variants coming up. And also it sort of really sparked a rethink about how we gather data and how much data we need in these spaces. If we can get the same or more signal with a single question than a in-depth 2 or 3 hour interview, then even though there might be a little bit more noise in the 1 or 2 question approach, it can get to many more people. And so that's one of the key things that we've sort of worked on in psychiatric genetics coming from these findings. So if we roll forward again, 2019, and another big jump in sample size, but another massive increase in the number of signals being found. And just to put these all kind of line them up, here we have, going across as you add more signal, more people, you get more signal. And the most recent analyses, which are just accepted today, was the notification email I got, have around 400 GWAS findings for depression. So it's definitely been a growing story, more signal, and that prediction is getting better and better as we have larger discovery samples. Okay, bipolar is the other one I'm going to talk about. Very similar situation, except already from that early stage where we had only around 18,000 people, we were actually seeing a couple of significant hits. So we do have a couple of loci in there that are slightly larger effect, but definitely not large effect loci. So the next jump brought the sample sizes up to about 50,000 people. And here there were 30 loci being found. The next jump brings it up again. There weren't that many cases added, but there were a lot of controls being added. And once again, this brought up the number of hits. And there is once again a very recent paper, which brings the number of hits up to about 400. And so what we're seeing is it's important to add screened controls. It's important to add cases. And as we add more people, it really is becoming very apparent how polygenic it is. So there will be more and more to be found. And when we look at the polygenic risk scores, we're actually looking all the way down the distribution. So we don't stop adding things to our predictor when we get to our significance line. We actually go all the way down the distribution. And we model that in a Bayesian framework so that we are sort of accounting for the noise in the space. But it is the case that as we add more power, things are going to keep popping up. And that's going to be really important when we think about how we use polygenic risk scores in clinic. Okay, so in terms of increasing sample size being a major factor to do better work in this space, we all had to have a bit of a rethink about how we were going to go about this. There's only so many 23andMes in the world, and no one research funder is going to fund recruitment or genotyping for that many people. So we do still need the individual groups across the many countries to be collecting data, but we can do it leveraging on that idea that rather than going through a clinic, we can actually maybe recruit directly from the public, and we can do it using a fairly sort of minimal approach to recruitment, but then collect a lot of in-depth information once we have recruited those people. So this is the approach that we've taken in Australia. This is Nick Martin, who leads this cohort. It's the Australian Genetics of Depression study. And basically, most of this cohort was recruited through a big media push. So there was a media release and a lot of sort of advertising around the fact that there was a study that people could come into. And the tagline really to come into the study was, have you been treated for clinical depression? So we say treated rather than diagnosed because people don't necessarily agree with their diagnosis, but treated is what we were looking for. Okay. So one of the questions when we do this kind of thing that we automatically get asked is, how do you know you've actually got people with depression? How do you know that these people aren't just having, you know, a bad day or a minor sadness rather than actual depression? And how do you know you'll actually get clinical cases? So along the way, we put a lot of effort into tracking the number of symptoms people were endorsing, and almost everyone was endorsing all of the symptoms of depression. We were also tracking the number of episodes. And in the questionnaire, we actually said 12 or above, which is why we have a very large spike here at 13, because many people had had a lot of ongoing depression or many episodes. And so this was reflected in the average age of our sample, which was about 45 at the time of recruitment. In addition to doing a really long online interview, the other part of this is we also asked for permission to do record linkage with prescription records, which in Australia are subsidised through a scheme called Medicare. And so we were also able to validate people's self-reports of having had depression, having been prescribed antidepressants and these kind of things. So we really don't think there's very much, if any, malingering in the sample or people coming in who don't actually have a diagnosis. They get no reimbursement. So the idea that someone's going to sort of spend an hour or two filling out a rather boring questionnaire and then also send in a saliva sample and get nothing in return, it really doesn't make sense that people would be faking this. And so looking at the genetic results we got from this sample, we can also show that actually, you know, these are the right people. So we, here I'm showing the prediction, the polygenic score results predicting depression in this cohort. We've got a strong prediction. We're explaining about 5.5% of the variation in this cohort based on this would be the Ray GWAS that we're using here. And we also have a strong SNP heritability. So there's quite a lot of sort of evidence coming together that what we have recruited is actually severe depression. And when we look in our cohort, the people who are in the top decile of risk for the polygenic score are actually 6.5 times more likely to have had depression than those in the lowest. So this includes some controls in this analysis, just to make that clear. Okay, so we've done a series of follow-up studies. We looked at how these people traveled through COVID. We've also been able to do some really interesting work about looking at diversity in this study and experiences of side effects and pharmacogenomics as well. And we've taken a similar approach to also recruit a cohort of people living with bipolar disorder, ADHD, autism. We've also done this with Parkinson's and also with schizophrenia. So it's been done pretty widely, and many of us have used this method now. All right, so that's kind of where things are at, and I'm going to be talking about those two cohorts again in just a minute. So there was a reason to introduce them. So I'm just going to sort of spend a few minutes talking about polygenic risk scores and mental health and how things might be a bit different in the mental health space. So one of the things you kind of get a feel for when you go to conferences and you talk to people who are doing the work in this space is that most people don't have any clinical experience or training in mental health. And that's not necessarily a bad thing, but it just leads to a couple of assumptions or misunderstandings that are sort of carry-ons from other spaces in medicine. And so the first one is that in mental health, the diagnostic systems are not actually based on etiology. So in other parts of medicine, those diagnostic systems are, or at least are meant to be, based on etiology. But in mental health, that's not the case. They're meant to be based on presentation and on symptom level data. And so I'm not sure I can think of a single depression genetic study that doesn't hand-wring about heterogeneity. Oh, it's so terrible. These people are so heterogeneous. You know, this makes our life really hard. Wow, this diagnosis is really bad. But the thing is, there's no expectation they should be homogeneous because the diagnostic system is not actually built that way. And it's not meant to be. And so that's one of the key points I think that's quite different in mental health. And also there's an assumption that clinicians actually want a yes or no answer. And that's also very seldom the case in the mental health space. What's going on is very complex generally with people. There's a lot of environmental input to mental health presentations, and working with the way that we design treatment and recovery needs to take that into account. And so most clinicians are not actually looking for a yes-no diagnosis answer, which unfortunately is what we optimize for when we're doing a polygenic risk score type study. And so because there's this assumption that they want a yes-no answer, there's a real focus on predicting cases from screened controls, but that's not really the issue in mental health. And it's also assumed that once someone has a diagnosis, it should be static over time or not change that much. But once again, in mental health, we expect things to change, and actually they should. Another major issue in mental health is that comorbidity or co-occurrence of conditions is extremely common. It's the norm rather than the exception. And so what I think is that rather than going for a straightforward polygenic risk score type approach, what we need to be thinking about in this space is thinking about profiles, because that's actually the way that a lot of psychology and psychiatry operates, and the way that a lot of clinicians are sort of taught to think or are comfortable thinking. Okay, and I'll unpack that in just a minute. So when we do polygenic risk score analyses, most of what we're doing is trying to maximize the variance explained or the prediction. And that's a completely valid goal. It's just not necessarily the only clinical goal out there. So this might work particularly well in cardiovascular disease or cancer, where knowing someone has a risk early means that changes can be made and interventions can be made. In the mental health space, this is a little bit more problematic just because there is so much genetic correlation between the different diagnoses. And so knowing about someone's risk for disorder A actually tells you a lot about all of their risk. And so this idea that there could be a targeted intervention becomes really quite difficult. Okay, so when someone makes it to a mental health clinic, they've been through a waiting list. They're usually having to pay. And it's not really a question of if this person is a case or not. They are. If they've made it through the waiting list, they've turned up and they're paying, something's going on. They're in distress. So the question is not usually, are they a case, but what type of case, or can we use this to help with differential diagnosis? And that's maybe where we need to be focusing rather than straight case control type prediction. One of the other issues is, one of our selling points about polygenic risk scores is we can genotype at birth or very early, and then that stays the same during the lifespan in terms of risk. But what's actually happening in the literature is about every 6 months we get a large improvement in our prediction, either through a new GWAS or a new method. And so those scores are actually constantly changing at the moment. And so if we're going to be using these scores in clinical practice, we need to think about what that means when, someone comes in January and their risk is 20%. Someone comes back in June and suddenly their risk is 25%. And then next January it's 15%. How do we have this conversation with clinicians? How do we have this conversation with patients? How do we make this meaningful? Okay, and then the next big complication in the mental health space is that the diagnoses are used for much more than medicine. So they go into almost every single court case there is across all jurisdictions and across different levels of courts. And they're also used to allow access or to deny access to a whole series of social supports. So if we are going to be suggesting that these things should be part of a diagnosis or could be part of a diagnosis, we need to think about the legal and ethical implications of what happens 6 months later when that changes and that person has to be reassessed. And so there's a couple of other bits and pieces we really need for diagnosis. One is that it needs to be usable in a low resource setting. Arguably, the cost has come down so much that in a Western medicine setting, it probably is usable, but that's not true across the range of services that are out there in Australia or New Zealand. And we also really need diagnostic processes to be measurement invariant. And so that's the idea that it will predict equally well for the majority of the population or all the population ideally. And we already know that's not true. So we know that the ancestral, the difference in accuracy between ancestral groups is a real problem here, but also the accuracy changes with the age of the person and potentially some cultural, environmental things that are also going on with the person. Okay, so just to kind of point out where the headspace is at and where I think we've got a few problems, and end on a positive note of where I think we could go. I've run some analyses that I'm going to show you around predicting depression or distinguishing depression from bipolar disorder, which is actually a major clinical question in the field. The data the data is coming from the Genetics of Depression study and Genetics of Bipolar study. And the polygenic risk scores I'm going to be showing were built from the psychiatric genomics consortium work. And I've built these using SBASE-R, so a Bayesian polygenic score method, which I can talk about as well. So I've deliberately done some naive analyses here and thought about the way that things actually would be used in a clinic. Okay, so the first question is the idea of first past the post. So which is your highest PRS? So here we have a group of people with a diagnosis of depression who have never had a diagnosis of bipolar or a manic episode. And we have a group of people who have a bipolar diagnosis who have both depression and mania. And what we can see is, although there is in terms of the numbers, it is statistically better. More people with depression have a higher depression PRS. We would get it wrong about half the time if we were to take this forward to a clinic. If we add the extra wrinkle of adding in some red herrings, so here I'm adding in a schizophrenia PRS, although none of these people have a diagnosis of schizophrenia or any psychosis symptoms, what we can see is around a quarter of these people, their highest polygenic risk score is actually for schizophrenia rather than depression or bipolar. And this is coming from the fact that there's a really high genetic correlation between all of these disorders. And so the idea that we could look at a person's polygenic risk scores across a range of traits and say, well, this one's highest, that's probably what you've got, it just doesn't work. The next thing, and this is something that's often done in clinical practice, is to say, well, it's a distribution, but only that top 10% really are clinically meaningful, and we'll take that. Sorry, 5% I've used in this analysis. It works the same regardless of where you set that threshold. Once again, this doesn't really work. So taking our group of people who have a bipolar diagnosis, only 13.2% of those people actually meet that top 5% threshold. But 10% of the people with depression who have never had a bipolar diagnosis or any mania also meet that threshold. And then same thing, vice versa with depression. So that clinical threshold idea, which is used for a whole range of stuff, also doesn't work. But other ways that we actually look at things in the clinic is through profiles. So profile type analyses and thinking is very, very common in mental health. So on the one side here, we've got IQ tests, and this is the way that people actually look at them in clinical practice. We don't just look at a number, we look at patterns of strengths and weaknesses. And the other one is actually a personality test. So this is a clinical personality test as opposed to a research personality test. And it's always looked at as a profile. Actually, the P in that personality test is really around profile and personality. So if we take that idea forward to polygenic risk scores, we can start to look at statistical differences between groups and test for equivalences in profiles. And this will be easier if I just show you. So in the green here, we have the participants who have a diagnosis of depression. In the blue, we have the participants who have a diagnosis of bipolar disorder. On this plot, we're showing 3 different polygenic risk scores, and this is the mean polygenic risk score for each group. What you can see is you start to see separation. So you can start to see some patterns, and they are statistically different from each other. But as we add in more and more of these polygenic risks, we'll start to see better separation. And we have some work underway at the moment that I have funding for to go out to groups of people with lived experience and go out to clinicians, ask them what type of things they would like to be putting into a profile if we were going to make something like this, and then designing an interface which would let people do that. So the idea is that this isn't, we're not targeting diagnosis here. What we're doing is targeting treatment planning and trajectory for a person. So we'll be including things like insomnia, weight gain, and so on. And so with the idea that knowing someone has a really high risk, for example, for obesity, a genetic risk, you know, that might change the way that you prescribe, or you might prescribe an adjunct weight loss medication already, or prescribe and subsidize exercise or something like that. So there's ways to kind of incorporate this that will help let us use genetics and polygenic scores in a clinical setting that have to do with treatment. So that's kind of where we're at, and I'll stop sharing and I'm happy to take any questions.